Thank you so much for joining us today, either morning or evening, depending on where you're calling from. We're excited to have you here. This is part of the Emory Alumni Association webinar series. We're delighted to have you for our third part in our Virus Without Borders series, sponsored by the India Alumni Network. This has been a great way to connect with our India alum and international alumni all over the globe on this very pertinent global pandemic topic that we're all living with right now. We're excited to be joined by three wonderful Emory experts to give you a little bit of look into their research and what is going on right now during the global pandemic. Two pieces of housekeeping. This is being recorded, so if for any reason you need to step away from your device, we will send you the full recording within 48 hours on our YouTube channel. And secondly, this is a live presentation, so if you have any questions throughout the time, please chat your questions in the question box, and I'll be monitoring those, and we'll ask those to the presenters with as much time as we have after they have their discussion. With that, I'm going to hand it over to our wonderful moderator, Nikhil Singla, who is going to be moderating this conversation and introducing our presenters. With that, welcome everyone, and thank you, Nikhil.
Great, thank you so much, Lane. And sincere thanks to Chris, Katie, Michelle, and the team for making this happen. A very warm welcome to everyone to our third webinar in the series, A Virus Without Borders. Today's discussion is focused on getting insights into ongoing cutting-edge research efforts to overcome the COVID-19 pandemic. Before we get started with the session today, a quick introduction to Emory's India entity. This legal entity announces Emory's formal and official presence in India. As you know, Indian students form one of the largest groups on Emory campuses. A large number of alumni are spread across major cities in India. A number of accomplished faculty members across different disciplines have research collaborations with Indian partners. One such exemplary partnership is between the Emory Vaccine Center and a premier research institution in Delhi. The efforts here are focused on infectious diseases research, including that on COVID-19. Building on these various efforts and parameters, the focus of the new Emory entity is to strengthen and expand Emory's footprint in India. And with this, I would like to extend a very warm welcome to our dynamic panelists today, who are rock stars in their respective areas of research and have been making seminal contributions in infectious diseases research. Dr. Mehul Suttar, Dr. Rama Rao Amara, Dr. Jens Remmert, thank you so much for joining us today.
Thank you.
Before we get started with our discussion, I would kindly request each one of you to give a brief introduction of yourself and a quick overview of your research interests. Mehul, maybe we can start with you, please.
Sure. So my name is, as Nikhil said, Mehul Suttar. I'm an assistant professor in the Department of Pediatrics, as well as a member within the Emory Vaccine Center. My research is mainly focused on emerging viral infections over the past several years and the subsequent host response to those viral infections in humans. We've studied various different viruses over the years, including flaviviruses and alphaviruses, and now this virus, this coronavirus, SARS-CoV-2. Our research is mainly focused on a couple of different areas. The first is looking very closely at the very early immune response after a virus infects a cell. So looking at the very first set of reactions that occur within a cell following a viral infection, like type 1 interferon induction, and the interplay between the virus and this innate immune response. We're also very interested in understanding antibody virus interactions and how antibodies can block the virus in infecting a cell. And we've developed several different assays to be able to evaluate the quality of these antibody responses in terms of its ability to block the virus.
Lovely. Thank you so much, Mehul. We are looking forward to getting more insights from your experiences in viral disease management on this pandemic. Rama, can I request you to please tell a little bit about yourself?
Hi, I'm Rama Amara, the professor at Emory University. I have multiple affiliations. My primary appointment is with, and my lab is also located at Yerkes National Primate Research Center. I am affiliated with Microbiology and Immunology Department in the School of Medicine and with the Emory Vaccine Center. My lab focuses on vaccine development. We started out with vaccine development and immunotherapy for HIV 20 years ago. I played a big role in taking one of the vaccines, that is MVA, DNA and modified vaccine, Ankara-based vaccine, into clinic. This vaccine has completed phase 2A studies in the US. The next step is to go into the efficacy studies. My lab works with various different kinds of vaccines, including the DNA and MVA. More recently, we started working on vaccines for TB and SARS-CoV-2. And we extensively use the animal models, mostly in non-human primates, and then we have done a lot of human studies. And we are actually working to develop vaccines for COVID-19 right now and making a great progress.
Great, thank you so much, Rama. We're really looking forward to hearing your insights. As we all know, vaccine remains a critical tool in addressing this pandemic. So thank you so much. Jens, can I request you to please tell a little bit about yourself?
Sure. Thank you for the invitation for this as well. So my name is Jens Rammert. I'm also assistant professor in the Department of Pediatrics here at Emory. My main research interest lies within humoral immunity or antibody responses to a number of different pathogens. We've done a lot of work over the years on influenza, but also emerging infectious diseases such as dengue and Zika, and more recently SARS-CoV-2. And so we study the development and maintenance of humoral immunity, both at a serological level, but also using single cell approaches. And this latter includes human monoclonal antibodies that have been developed both as a means to understand the fine detail of immune responses, but also providing reagents that can be used both diagnostically and therapeutically against these various diseases.
Great, thank you, Jens. We're also, looking forward to hearing about these approaches that you mentioned as we wait for an effective vaccine for addressing this pandemic. So thank you so much again to the three of you. This would be an interesting informational discussion, I'm sure, for the audience, but also for me as we, sort of get this plethora of information on the different things that are happening to control this pandemic. So your detailed scientific insights would be very useful in understanding what progress we are making against this, against the SARS-CoV-2 and addressal of this pandemic. So as we know, this pandemic has now infected over 7 million people, has resulted in over 400,000 deaths, and is also affecting specific groups more strongly, like the elderly are more vulnerable to this disease. Maybe my first question in this context will be to you, given your extensive experience in understanding and management of viral infections, what about this virus is different in terms of how it attacks the host? As we have seen it, it has a selective preference for different groups, and it's been very contagious. So what is it about this virus that gives it these different features and makes it this challenging health issue that it has become right now?
Thank you, Nikhil. So there's this SARS Coronavirus 2, which is name of the virus that causes COVID-19, is one of the third major iterations of one of the major epidemic coronaviruses that has caused significant public health concern over the past 2 decades, I would say. So SARS coronavirus emerged in 2004, and this was a virus that was originally found in bats that went to ferrets, and then eventually crossed over to humans. This virus is one of those cousin viruses to the current pandemic virus, SARS-CoV-2. There's also another virus called MERS that has circulated within the Middle East for the past 5 or 6 years. This virus has a high mortality rate, much higher than what we see with SARS-CoV-2, but this virus is slightly different than the virus that's currently circulating. SARS-CoV-2, there were a couple of very important discoveries made very early on in terms of how this virus spreads. Certainly, this virus spreads through respiratory droplets. People can spread it by coughing, sneezing, not having proper hand washing techniques. But also, one of the major impacts that has really affected everyday life is this notion that this virus also spreads through asymptomatic individuals. So this virus, for many different viruses, symptomatic individuals are the ones that can spread these respiratory viruses. But when it was found early on that asymptomatic individuals, people that don't really know that they have COVID-19 infection, may also be individuals that spread this virus. And then one of the other major findings that was made, similar to what was made with SARS, is that there seems to be some sort of age-dependent effect with this viral infection. Young children don't seem to be as well affected as compared to the elderly. And then there's also people with pre-existing conditions, whether it be heart disease, type 1, type 2 diabetes, maybe even some pre-existing lung conditions, seem to be more affected by SARS-CoV-2 infection. It's unclear precisely why the elderly or people with pre-existing conditions seem to be worse off following infection. I think this is an active area of investigation by a number of groups across the US as well as across the world, I would say. And this is, I think, something that's going to be really important to understanding how we combat this virus as well. I think Rama and Jens will probably touch on this as well, is that when you're trying to drive a vaccine response, you want to be able to protect the elderly as well as those individuals that are maybe immunocompromised or have pre-existing conditions. Those are very challenging in terms of driving an immune response in those individuals. Again, the mechanisms and the reasons for why it's more difficult in those individuals is still an area of active investigation. So when this virus spreads through a respiratory droplet, one of the places where the virus immediately infects is the lung cells. It interacts with its receptor, this lock and key mechanism where the virus interacts with the receptor called the ACE2 receptor that's found on many different lung cells. And from there, the infection progresses. One of the huge distinctions between SARS-CoV many years ago when that first emerged and this virus is that this SARS-CoV-2 virus seems to have a much stronger affinity for this ACE2 receptor than SARS. So thus it's able to infect cells and cause disease, as well as spread in asymptomatic individuals, like we mentioned earlier.
Great. Thank you, Mehul. This is interesting. I guess we don't yet have the structural analysis of this interaction as well to get us more insight. Is that correct?
So people have done some cryo-EM structure of the spike protein, which is found on the outside of SARS-CoV-2. It's those little spike-like projections that you see outside the viral particle. And people have also modeled the ACE2 receptor. Again, this interaction between the spike and ACE2 has been studied in the context of that older and more extinct virus, SARS coronavirus. And so a lot of the tools in that context were already developed. So when SARS-CoV-2 came along, that interaction was already well understood from the fundamental basic interactions of the spike protein with this receptor. And so groups early on were able to quickly model and generate structural diagrams of what the spike protein is, the receptor binding domain, which we'll touch on much later, and how that interacts with its receptor, the ACE2 receptor.
Okay, great. Okay. Thank you, Mehul. As we are speaking about these novel parameters of host-pathogen interaction, I would like to just shift gears a little and move towards the vaccine design and development approaches that are ongoing. And before I speak to Rama about the specifics of those approaches, I would like to request Jens to give us a little bit of an overview from your experience in vaccine design. Like what are the major steps that are involved in designing a vaccine for a novel pathogen such as the SARS-CoV-2? Given the challenges that we are facing and the novelty in its host-pathogen interaction that we are seeing, what are the major, sort of designing steps and approaches towards developing a vaccine for such a pathogen?
Right. So for most vaccines, of course, what the primary points of importance is to generate a vaccine with both a very strong immune response, but also a durable immune response. We would prefer to see a vaccine that provides long-term protection as opposed to something that dissipates quickly. But I think in this particular case, as Mehul alluded to, some of the concerns is that the groups of people that we want to protect the most here is also traditionally difficult targets to get an effective vaccine in. The elderly, immunocompromised patients, patients with cancer, for example. And so I think while these early efforts are really focused on generating a vaccine, as we go forward, I think all these things would need to be taken into consideration as well. And then, of course, the other aspect that I'm sure Rama will touch more on as well is what component of both the immune response do we target with these vaccines, T cell responses, antibody responses, and so forth. And in doing so, what parts of the virus will we focus on in designing these vaccines?
Right. Thank you, Jens. On that note, Rama, I would request you to provide, given your extensive experience with the HIV vaccine design and your current work on COVID-19, 2 bits. One, any cross learnings in terms of vaccine development from an HIV perspective to now handling this difficult virus, the SARS-CoV-2, and the specifics. You alluded to certain approaches that your lab is taking in terms of candidate vaccines for COVID-19. So if you could highlight also what are these different approaches based on the issues that Jens highlighted, and the current status of these vaccines, where do we stand with them? We keep hearing about a number of different candidates from the US, from China, and across the world. So if you can give us a quick overview of how do these difficult viruses compare in terms of vaccine design to what's happening in your lab, and thirdly, where do we stand globally in terms of these different candidates and approaches?
Yes, there's a lot to cover there. I'll try to break it down. So first, I would like to say that one of the best thing that happened, that all the investment that the scientific community and the funders made into HIV vaccine development, I feel that they're playing a big role here in generating vaccines rapidly for COVID-19. At least, it's paying off because we understand a lot now about how to basically drive a specific immune response that we want. And as Jens pointed out, that one is the safety of the vaccine is the paramount. Even if you don't generate a protective immunity, at least you don't want to generate something that is not safe, or rather, and also doesn't enhance infection. The safety comes first. And the second is the magnitude of the immune response, and then how long it persists. So this is the key, because when you try to vaccinate people, trying to get them back to vaccination is a very hard task. It's people don't have time and to come back and getting additional boosters. So it is very important to have a vaccine that can induce a durable immune response, especially with a single shot. If you have a vaccine that can be achieved, vaccination can be achieved with single shot, that's the best. And also the immune response should be induced pretty rapidly, so that you can quickly vaccinate people and get them the protective immunity. So so this is, I think, inducing an immune response that can protect against the virus is not the real challenge. I feel that we can easily get there, but generating a response that can be induced by one shot, or at the most 2 shots, and then having that response persistent for a long time, that's what is going to be the key. And we should also pay attention to the future outbreaks. So we had SARS 1 in 2002, and then we had MERS, then now we have SARS 2. So if we design vaccines that can potentially protect against future SARS viruses, that will prepare us, better. So I think the vaccine development should really focus on all these aspects. So you have the safety, the magnitude, durability, and cross protection, right? So and okay, so now that's the major goal, the way I see it from a distance. So when we are trying to do this, so what kind of immune responses that are going to be protective? So I think the antibodies, everybody understands, are key. And generating an antibody response is against, for example, the spike protein of the virus is not that hard. We learned that it is very immunogenic, means it would induce a pretty strong immune response. I was actually pleasantly surprised to see it is so super immunogenic compared to what we have been doing with HIV envelope. To see a good antibody, binding antibody, this is just that binds to the virus protein. We need at least 2 shots for HIV envelope. Whereas with this particular protein from this COVID-19 virus, even the single shot can induce a pretty strong immune response. So when I saw that first, I was like, wow, this is, for the past 20 years, we have been struggling so hard to induce a strong antibody response, binding antibody response to HIV envelope. We go to an extent of using adjuvants. These are immune potentiating agents. That we had to use very strong adjuvants. As the strength of these adjuvants increases, the safety becomes an issue. Unless we do that, we were not able to generate strong antibody response. Whereas with this protein, it's easy. It's relatively easy. I would say this is 100 times maybe more immunogenic. So that's a good thing. But then the question is that, are we inducing these antibodies that we are inducing, can they actually neutralize the virus? So the neutralizing means that it can block the virus entry into the target cells. And what we are finding is it depends on what part of the virus that you use as an immunogen put into your vaccine. And you can induce a pretty strong binding antibody. These are the antibodies that bind to various sites on the, let's say, spike protein. But unless they bind to the business end of that protein, where the protein interacts with the host cell, they don't neutralize. So to generate an antibody that focuses on the business end is what is key. So that's where the challenge is going to be, that how you select your immunogen to target that business end. And there is also a potential that if you actually don't target the business end, target other parts of the protein, you could potentially enhance infection. So that's where the vaccine-induced enhancement of infection comes into play. So we need to be very careful before we run too fast into the clinic. We carefully looked at what is the vaccine is inducing, not just binding antibody, for the quality of the antibody in terms of blocking infection and then not enhancing infection. So I think we made a significant progress in that direction. And the second part is the T cells. So these are the cells that you have helper cells, you have killer cells, and these helper cells help both these killer T cells as well as the antibody production. So these killer cells, what they do is they clear, if the virus gets into the host cell, they clear those cells. So and the nice thing about that is that there is a lot of conservation in the sense that cells that can see COVID-19 virus very likely to see a lot of other SARS viruses. So that gives an advantage of a vaccine that is inducing both antibody and killer cells that can actually prepare you for protection against a lot of future other SARS infections. So this is basically what you need for the vaccine development. So then now if you look at, there are like over 140 vaccines being developed, right, across the world. That's only what is reported, and there are a lot of people have not reported that they're working on vaccine development. For example, my lab hasn't really been advertising that we are developing vaccines. We are doing these things quietly and very fast. So there are vaccines that are already into clinic, well into getting into phase 2 studies. So you have different kind of vaccines. You have mRNA vaccine, that is Moderna's vaccine. You have viral vector vaccines that's coming from Oxford and J&J and through Harvard. You have the DNA vaccines that Emory is doing, and there are protein-based vaccines. There's so many different companies or different people are taking different approaches, right? So most of these target the induction of antibody, but when you combine the more than one of these, that's when you get both antibodies as well as T cells. So I'm this is a very vast topic. I might have already taken a lot of time, but I basically like to conclude is that the safety is important. If it can induce immune responses with a fewer doses, 1 or 2 doses, that is very important. That immune response should be able to persist for a long time. If you can target both antibodies and T cells, that would be very, very important. That can give cross protection. So I think I'll stop there and let other people talk.
No, please, thank you, Rama. This is, the vaccine, the design for it, and when will it come are, the most, are questions which are on top of everyone's minds as different countries battle. And as you know, India is, with the increasing number of cases that are happening here after the lockdown has been lifted and what we're now calling the unlock. So this is good information to know. Just a quick follow-up on that, Rama. Like, what, according to you, are certain features of the SARS-CoV-2 that you feel are aiding in vaccine design on the parameters that you just outlined? Including, like you said, a good antibody response compared to vaccines being designed for HIV, and what
So Nikhil,
I think I think you're breaking up a little bit.
Anyway, so I think I understood part of your question. So I also would like to highlight that the SARS-1 infection and the vaccine development against SARS-1 actually helped us in a big way. That is that we know that the SARS-1 spike protein is quite similar to the SARS-2 spike protein. And we learned a lot about which are the regions of that protein to target. And that there have been vaccines that have been developed. They were tested in different models, and then their protection was actually shown. So because of this, a lot of vaccine developers could actually start vaccine development rapidly for SARS-2 without having any animal data. So people went straight into humans because of what they learned with the SARS-1, because they had to take that risk of making a mistake. But the chances of success was going to be very high because of what was known against SARS-1. I think that really helped fast track the vaccine development. And honestly, that a lot of the vaccines that are now getting into phase 2 now, those are designed based on what they achieved with the same platforms against SARS-1.
Please. Thank you, Rama. Sorry, I lost my connection in between a little. So I guess we will address any vaccine-related questions towards the end. In the interest of time, we will just shift gears again a little here. So I think there have been 3 major approaches as we are trying to contain this pandemic. With testing, tracing, and treatment. Like, when we have spoken about treatment from a vaccine perspective briefly now, would like to move to the first pillar again about testing. And in this regard, Mehul, again would like to get some insights from you about the different approaches that we have been hearing about, the different testing and diagnostics approaches that are being used for COVID-19 and which is playing a major critical role. From a public health perspective, there is the number of tests, but from the scientific perspective, there is the type of test that we are using. Could you give us an overview about the different tests that are in the market now, the pros and cons of each one of them, and how affordability, accessibility, and the precision of these different tests is playing a role in getting a handle over this pandemic?
Sure. Thank you, Nikhil. So there's two major types of tests that are currently available for diagnosing and identifying patients that are infected with SARS-CoV-2. On one side, you have a nucleic acid test, which is basically what I think is more commonly referred to as a PCR test in the public. This is a test where they look within your nasal cavity by doing a nasal swab, and then performing what's called the reverse transcriptase PCR reaction to try to identify if the viral nucleic acid is present within your nasal cavity, within your respiratory compartment. One of the pros with this test is that it's rapid, it's highly specific. So if you have a nice primer probe set, which I think both the CDC, WHO, and several other groups have developed, you can rapidly identify viral infection within patients within a few hours. These tests tend to be more costly, but also I think what's most important is that the window for identifying patients that are infected is relatively small, right? So if you have an infection, an active virus replication within your nasal cavity or your respiratory compartment, you can detect it. You can actively detect virus infection within this compartment by using a nucleic acid test. However, this doesn't really address patients that may be or individuals that are asymptomatic or mildly symptomatic, or people that want to know, have I been exposed to this virus or not? And that's where serology tests have been pushed on the market over the past month and a half. We've seen a number of different serology tests that have been developed over the past couple of months. One which I'd like to highlight is from Jens Rammert's group here at Emory, which helped develop a highly specific serology test that's specific to the spike protein, and in particular, within that spike protein, what we like to refer to as the Achilles heel, the receptor binding domain, the portion of the spike protein that actually interacts with the ACE2 receptor. His laboratory was able to quickly develop a serology assay that can determine whether infected patients have been exposed to SARS-CoV-2. And in my lab, so there's does the virus, have you generated an antibody response to the virus that can react to the spike protein of this virus, the receptor binding domain? What my laboratory has done is to help develop a couple of different high throughput based neutralization assays. And what neutralization assays are is looking more at the quality of that antibody response. So looking at, have you generated an antibody response, and how well of an antibody response have you generated? And I think Rama had touched on this in the context of generating a potent vaccine. So these neutralization assays combine the actual virus with a patient's antibody or serum or plasma, and you combine it and ask how well do those antibodies block the virus from infecting a cell. So you can both look at the quantity by an ELISA-based assay, as well as a neutralization assay to look at the quality of this antibody response. And as mentioned, there's also a nucleic acid test. There's many different types of serology tests that are currently available on the market. there's many pros and cons to these different serology tests. One of which is the nucleocapsid antibody test that has been pushed on the market. This just tells whether there's an antibody response to a particular viral antigen, which is more on the inside of that virus particle called the nucleocapsid protein. Doesn't really tell you much about whether you have generated a neutralizing antibody response. And then there's several other spike protein-based serology tests, and they vary dramatically in their level of sensitivity and specificity. What does all this mean to the general public? This means when I take this test, how reliable is it that I'm getting the correct answer over and over and over every time this test is administered? And these tests have been shown to have a wide variety of false positive rates or answers that may not be correct when you run through these diagnostic tests. They've varied as low as 5 to 10% false positive rates, all the way to as high as 50% false positive rates. So you have to look at the quality of this serology test that was developed, whether it's in the US, whether it's approved through the Food and Drug Administration, the FDA, and how specific and sensitive is this test. The one that the Rammert lab has developed is highly specific and highly sensitive, and it shows a strong correlation with how strong of a neutralizing antibody response there might be as well. So I think those are some of the advantages of the test that the Rammert lab has developed.
Thank you, Mehul. I think, yeah, thank you, Mehul. And I think on the note that you left on this, the antibody response, the antibody generation, the neutralization, on that note, Jens, and given in the interest of time, I would like to get your insights into these alternative approaches that we have been thinking about while we wait for the vaccine. Looking at plasma therapy, looking at monoclonal antibodies. Could you tell us a little bit about these approaches that the general public may not have heard about before, from a context of treatment and solutions for diseases as much as we have heard about vaccines? What do they mean in the context of addressing the COVID-19 pandemic, and how effective and useful could these be in controlling this pandemic?
Right. So let me start with plasma therapy and then move on to monoclonals after that. So using immune sera is really an age-old approach that's been used for a very long time. And the basic concept here is that you can harvest plasma from a previously infected and essentially a survivor of the disease, and use that as a therapy for very ill patients to treat the disease, right? And on paper, this seems relatively easy and straightforward, but it's more complex than you would imagine in terms of harvesting the correct plasma, etc. I think the main point here, and this is something that Mehul and I have been involved with here at Emory, is really identifying the optimal patients to provide the plasma for therapeutic use. You really need to identify the patients with the highest neutralizing titers of the virus to use those, and from our own studies and many other groups as well, we see that patients who've survived an infection have a huge range of immune responses that range from very low to incredibly strong, right? And so we need to pick the correct donors for these efforts. And then the other issue is what the target population for these therapies should be. we tend to use these plasma therapies as a method of last resort, if you will, for the extremely sick patients in the hospitals. And in this particular disease, the pathology or the problems that the patients are seeing is not just derived from the virus itself, but also from the exorbitant activity of the immune response within the lung itself, right? And so when we treat with whether it's serum or plasma or monoclonals later on, it's very important kind of what time point we're treating with here as well. So I think those efforts are ongoing, certainly across the US, and I imagine worldwide as well. But I think what's missing there currently is sort of a standardization of what those therapeutic efforts are and what the material, what the quality of the material given should be. And so that's very much something in flux at this point, I think. So then the other point you mentioned was therapy using recombinant human monoclonal antibodies. And so this is really a field that has exploded over the last decade or so, with human monoclonals being used not just for infectious disease, but as you know, also for in the context of cancer therapy, modulation of autoimmune responses, etc., etc. And our toolkit, if you will, to generate these very quickly has really exploded in the last, even in the last several years, I would say. So several companies at this point and private institutions as well have generated highly effective monoclonal antibodies that targets and neutralizes SARS-CoV-2. Some of these have gone into clinical studies already and are being evaluated for therapeutic use. Again, I think the key point here is to define what are the characteristics of an antibody that would be useful for this. Certainly, what is the cost of such a therapy? As you can imagine, that would be quite expensive. And what is the optimal time point in the disease course where such an intervention can really make a big difference? And so even though this might be something that is more readily applied before a vaccine is developed, I think there's a lot of questions that are still remaining to be answered in this realm.
And in this context, Jens, are there any particular efforts in your lab in terms of technologies, platforms, approaches that are accelerating work in these two spheres, which seem to be highly critical?
So we've worked for I guess close to a decade now on these type of approaches, and we pioneered some early work on single cell isolation of human monoclonals. This, of course, now, as I mentioned, are approaches that are used worldwide by a number of labs and companies as well. And the development there is really ongoing. The ability to do this now very, very rapidly is nothing short of amazing. It's really the technical ability in this realm has been incredible. I think still in the realm of monoclonals, one of the caveats, if you will, is simply the cost of producing these recombinant proteins. In small scale, it's not a problem, but if we're envisioning a larger scale, it does become an issue. The other thing I wanted to mention in this realm is it also provides an opportunity to treat the very patients that we discussed before, or that may not be able to mount a good vaccine response, right? So this would be, again, in the elderly, immunosuppressed patients, patients on chemotherapy, the whole transplant population. All of those patients are traditionally very difficult to mount vaccine responses in. And so these passive immunization, as it's called, whether it's using plasma or monoclonal antibodies, would be patient groups that would be treatable with those approaches that not necessarily are easy to treat with a vaccine once we have it.
Right. No, thank you, Jens. Thank you so much for that. So we have 10 minutes left, and before I request Lane to get us with any questions that the audience may have, just very quickly, Rama, as we discussed vaccines, we discussed these different approaches using plasma therapies, therapeutic antibodies, and of course novel testing methodologies. Just from a vaccine and from an immunity perspective, wanted to get your insight into this feature of herd immunity. We have been hearing about it quite a bit. Different countries are using that as a different tool. We hear about that from what Sweden is doing, about what it could, how it may or may not work in India. So could you please tell the audience, what does this mean in simple language, and what are the different contexts to it from the perspective of COVID-19?
So herd immunity, I think, which is very important. It's in a simple language that when somebody is exposed, not everyone who is exposed is going to develop disease. So at exposure at low levels potentially could act like a vaccination, and these individuals induce an immune response against the virus, and because of that they will be protected. So this is very important, and I think one thing that we don't know yet is that if somebody is exposed to the virus once, and do they actually have protective immunity, or they actually, when they are exposed to the virus again, whether there's going to be enhanced infection. So that part, I think, needs to be thoroughly established before we can say what the herd immunity is going to do. And then second point there is that how long this immunity would last. That's another thing that we don't know fully yet. I think that's where both Mehul and Jens are also looking at carefully in these exposed and protected individuals or cleared individuals. So it's going to, I am sure in a few months we'll have more data in that direction. But then we also need to be prepared if people who are infected, we can actually treat them to be able to control the pandemic.
Right. Thank you so much, Rama. Lane, do we? I'll just leave it to you now for some questions from the audience.
Great, thank you, Nikhil. Thanks so much to the presenters. We've got a lot of really great questions that have been submitted. The first one asks, in what ways does the current attention on COVID-19 affect vaccination for other viruses that were already underway? Are these vaccination efforts now receiving less attention? And if so, are there certain parts of the world more threatened than others?
So yes, so Rama, I think we alluded to these points in our discussion, but if you could please highlight them again.
So it's a very good question. I think that we have to make sure to continue with the routine vaccinations that are very critical. In the short term, because if people are following these social distancing practices and all these clean sanitizing practices, I think the spread of other infections are also likely to come down. So in a short term, if there is a delay in these vaccinations, I don't think it's going to impact in a big way. But in the long run, we have to make sure that those will get done as well.
Thank you so much. This next question is for Dr. Suthar. If you had to recommend just one serology test to the general public, which one would it be? And could you talk some more about the test that the Rammert lab has developed?
Yeah, so I would recommend the test that the Rammert laboratory has developed. I think there's a couple advantages to this test. As I said, this test was generated very rapidly by his laboratory, but was also moved to the clinic at the Emory Medical Laboratories and was put to use within the clinic within a short amount of time. It's currently undergoing FDA testing to be able to achieve the type of CLIA certification that these tests require. But I think this test also tells you about, or can serve, I should say, as a surrogate for what is the type of quality of the antibody response that I have in terms of correlations to neutralization within these tests. I believe that the diagnostic test at Emory Medical Laboratories, this receptor binding domain test, has the capacity, and Jens can chime in here, to be able to screen about 5,000 or up to several thousand patient samples in a day. And they're currently optimizing it such that they can screen healthcare workers as well as the general public over the coming weeks.
Great.
Yeah, again, like we said earlier, I think the important thing here is to consider a diagnostic test can give you just that. Does it answer the question, was I exposed to the virus in the past? But the other aspect is, does it tell me anything about the quality of that immune response, i.e., am I likely to have a good neutralizing immune response? And looking at for example, an internal viral component, such as the capsid Mehul mentioned, it doesn't really provide you with that information. So I think as a consumer, it's important to understand what test is it that I'm getting ready to have administered, to understand what you're looking at.
Great, thanks. I think this is going to end up being our last question for the session. A former student of Jens and Rama, Rakas Ella, says hello, by the way, and wants to know, what is your opinion on the role of ADE for an inactivated COVID vaccine?
So this is what the issue that Rama has mentioned about the potential of seeing enhanced disease with the vaccine, and this is something that has been discussed at length in the field of dengue vaccines and to some degree with Zika vaccines as well. And so the basic premise is that you would have induced an immune response that actually does the opposite of what you want from a vaccine. It actually causes enhanced infection once you're exposed to the virus. And I think that's where the choice of the antigen comes in, as Rama explained before. There are parts of SARS-CoV-2 that is more conserved with seasonal coronaviruses than others, and I think choosing those parts that are not conserved would alleviate a lot of that concern. I don't think that there has been any data even suggesting that this is a major problem as of yet. Maybe Mehul or Rama, you can comment on that. I'm not aware of it.
Yeah, so I think one of the pieces of information that was that I was involved with many years ago is immunization against the nucleocapsid protein, the inner guts of that viral particle. It seems as though the nucleocapsid, the antibody response to the nucleocapsid protein may contribute to immune pathology, and that's something that's being looked at certainly very closely. That was observed in the context of SARS. So if you immunize mice with nucleocapsid protein and then challenge them with the virus, what they found was that mice that were immunized against the nucleocapsid protein had much greater lung pathology on a subsequent challenge as compared to naive mice, suggesting that the nucleocapsid protein may be contributing to some lung damage. There's some groups that are suggesting that this may be complement mediated. It's an arm of your immune response, an antibody related complement arm. But I think this is something that from a vaccine standpoint needs to be looked at much more closely. But certainly I think an antibody response to the nucleocapsid in general is not going to be protective because it's not found on the outside of the virus. It's not going to neutralize the virus or block the virus from infecting a cell. So I think most vaccine efforts have focused primarily on the spike protein and various elements within the spike protein, like the receptor binding domain.
Yeah, a comment on that. It's very important here to distinguish between antibodies actually causing pathology themselves. An alternative thought on what Mehul described here is by priming the immune system to nucleocapsids, you're also essentially going to infect the natural immune response to the infection itself later on, right? So the immune response that infection will now drive will be honed in on the nucleocapsid, since you were exposed to that before, and you will fail most likely to mount a good neutralizing antibody response because of it, right? So that could in part explain why the titers of the virus went up in that particular situation. Mm-hmm. Or it could be an immunopathology based off of the nucleocapsid specific antibodies. So as with many things, there are alternative interpretations of this. Yeah.
Okay. Thank you so much, Jens, Rama, and Mehul, for this informative and insightful discussion. I'll hand it over to Katie now for the closing remarks.
Actually, I'm going to close this out. I just want to thank our presenters so much. I know your time is incredibly valuable right now, and we really appreciate you sharing this great insight with our alumni networks. I just want to remind everyone who joined us to please come back for our June 18th presentation, which is going to be policy impacts on the health and safety of our two nations, India and the US. That's going to be in the recording and the email follow-up that's sent later today. Again, huge thanks to our presenter, Anton Nikhil Singla, for joining us and sharing this great information. And we look forward to seeing you soon. Take care and be well. Thank you.
Thanks for the opportunity.
Thank you.
Thank you.
Bye.